We have already talked about G protein linked or coupled receptors. Those are not the only receptors that play an important role in the biology of a cell. There's another class of receptors, the enzyme linked receptors, which play a very important role as well. The signals for these receptors generally specify growth, proliferation, differentiation, and cell survival. These are usually local mediators and generally very low concentrations. The response to these signals is in an amount of hours rather than seconds that we saw when we were talking about G protein coupled receptors. Generally, as response to these signals, there's a change in gene expression. The receptors for these signaling molecules or these ligands are single transmembrane proteins. It's a single polypeptide versus, as we saw in G protein coupled receptors, it was a protein that crossed membrane 7 times. We call it a serpentine receptors. The cytosolic domain of these receptors has an intrinsic enzyme activity or is associated with an enzyme. The extracellular domain, of course, is responsible for detecting the ligand or the signaling molecule to which it binds and causes a conformational change that affects the cytoplasmic domain as well. Some of the receptors and their signaling molecules are mentioned on the screen. Epidermal growth factor stimulates proliferation of various types of cells. Insulin stimulates carbohydrate utilization and protein synthesis. Insulin-like growth factors, they stimulate cell growth and survival. NGF, nerve growth factor, stimulates survival and growth of some neurons. Platelet-derived growth factor, PDGF, it also stimulates survival and growth and proliferation of various types of cells. You may notice that other than insulin, pretty much every signaling molecule, every other signaling molecule is involved in cell growth, proliferation, or survival. So these receptors basically can detect the signals, and the information they convey to the cell is regarding survival of the cell, differentiation, or growth of the cell. These receptors, although they are single polypeptide chain, in order for them to function or communicate the message which they receive via ligand to the rest of the cellular machinery, these receptors generally have to dimerize. What do I mean by that? The two receptors have to come together, and when they come together, their cytoplasmic domains, when they are sitting in close vicinity of each other, they have a weak kinase activity. When they come together, the kinase domain of these receptors, here I can point that out. Here's, for example, the kinase domain of this receptor. When these two domains come close together, they cross-phosphorylate each other, which means this kinase domain is going to phosphorylate different amino acid residues on this receptor, and this kinase domain is going to phosphorylate special amino acids on the other receptor. So when the ligand binds, the receptor dimerizes, there's a conformational change which causes enhancement of the kinase activity by phosphorylation. First of all, the kinase domain is phosphorylated. That increases the kinase activity of these molecules, and then that kinase domain phosphorylates rest of the other domains of this receptor. So how do we know that dimerization is important, but it is the kinase domain of these receptors that ultimately causes an effect in the cell? A simple experiment people have done is that they have in a cell, they have a normal receptor and they add a mutant receptor to the cell. This mutant receptor does not have the kinase domain, the domain which has the enzymatic activity. So if these mutant receptors are in abundance, they pair up with the normal receptors. However, they can pair up, they don't have the kinase domain, so they cannot phosphorylate each other. So when that happens, although the ligand has bound, there is a dimerization of the receptor, but there's no kinase activity, so there is no message conveyed to the cellular machinery. This is also termed as dominant negative inhibition. Remember, when we were talking about genetics, we said that genes that code for proteins that are responsible for enzymatic function, if there's a mutation in those genes, it results in a disease which is transmitted in a recessive form. Here we can see that even if one gene is normal, we have 50% receptors are normal, the other 50% are abnormal because the gene that coded them has a defect. This will result in a negative dominant scenario. Basically, although 50% of gene is normal, the half which is mutated will prevent from this receptor from functioning normally. Another type of receptor which is included in this category is afferent receptor. Afferent receptors, they regulate cell adhesion and repulsion responses, migration of the cells, and also axon development. These are the most numerous receptor tyrosine kinases. There's also a special thing about these receptors. There's receptor ligand, both of them are present embedded in the plasma membrane, and just as afferents signal the receptor also gives a signal to the ligand. So it is bidirectional reciprocal signaling. So the end result is either cells respond to such signals with cell adhesion or repulsion. So what determines whether a cell is going to adhere or repel? It is the machinery inside the cell that is going to interact with these receptors and convey the message to the other components of the cellular machinery. So let's see what happens when these receptors are activated and what proteins interact with these cytoplasmic domains of these receptors.